Shall we start?
Yeah, I don't see why not. Everybody's here.
Good idea.
Everyone we can reasonably expect.
So today I'm very glad to have the opportunity to introduce Keqiang Li, who joined our group last year and began to work on this project. So as some of you may know that this is a project that we have been working on for many, many years. So a graduate student, Gang Su, who did his thesis work in our lab, basically laid the foundation and doing kind of the proof of concept work for the CoolMap. And after Keqiang joined our group, he basically continued the development and now transformed the initial basic framework to a platform that we hope can greatly facilitate omics data exploration. So this actually will be the first time that Keqiang doing a presentation about a biological topic.
Yeah.
So he was.
I was a computer science guy. Yeah.
Let's see what he will say.
Yeah, what will happen? Yeah. Okay. Okay. First, thank you guys for coming. And I think some people were wondering about my name. For example, they might have seen it on some Chinese news or something like that. I think, yeah. Yeah. There's sometimes you search it on Google. I won't tell you why. But okay. Okay. So let's get started. So first of all, CoolMap is just a tool like to help researchers to better understand their data and like do navigations, do the aggregations, and do visualizations of the data, and help them to do some make the hypothesis or things like that. So this is kind of a major challenge for a lot of researchers. Daily life is to make sense of large data set. And there are two approaches that researchers use for this purpose. The first one is computational approaches. Computational approaches, the researchers always know what kind of model they're going to use to evaluate or what kind of hypothesis they have already made. So this always offers definitive answers to the result part of the paper. But there is another approach called interactive visualization approach. In this approach, it actually provides a global view of the data. So you can quickly assess your data and try to make the hypothesis. So which is very convenient for you to do the initial hypothesis development. So, but most commonly, researchers will combine those two approaches together to do the data research. So this talk will focus on actually more on the interactive visualization approach. One of the solutions is like this. So you just, this kind of a major barrier for researchers. Like you can't really fit all the data in the screens, the monitors you have here. So it's always impossible. Like, let's say for 20,000 genes and 1 pixel width for each gene, you need like 5 4K screens or monitors standing vertically here to show all the genes. And it's kind of not practical, if not impossible. So that's why we have CoolMap now. So basically, you can see here, I put this equation here, is CoolMap is equal to heat map plus dynamic row and column ontologies. So heat map is, I just assume everybody knows it because it's just a mapping from the values to the columns. But based on the heat map, we add this dynamic row and column ontologies to CoolMap, which is user can actually, CoolMap comes with this function that can dynamically expand and or collapse the row and column nodes. So this enables the condensation of large datasets into human manageable views. So user can examine the fine details in a very high level context. So CoolMap lets scientific users aggregate rows and columns into pre-existing concept groups, or like some pre-computed clusters. So, but in this way, scientists convert overwhelming data matrix just with huge amounts of data into a much smaller view. Okay, now let's take a closer look at how CoolMap functions. This is, I won't just read those texts, but you can see those two figures here. The first one is showing how CoolMap collapse the data, aggregate the data. So in these figures, we use mean aggregation. By collapsing those two column and those two row nodes each into one row node and column node, we get the aggregation value 2.5 for this. And the second figure shows how CoolMap expand the views of itself. So this node and this node both expand to two nodes, which now you can find like more detailed values here, while you still keep those high-level contexts here, like which parent node it belongs to. So this is basically how CoolMap display and manage data. So next, let's take a look at its core functions and the basic operations it can do. So from here, I kind of want to do some live demos of CoolMap. So just forgot to mention that all the core functions have been developed by Keqiang, who is my former colleague. I took over all the work from him. So all the core functions I will introduce here have all been done by him. So first of all, so every time you download CoolMap, it's on all the files comes in under a folder called CoolMap, and because CoolMap was developed by Java, so it's a Java file for now. So let's just start it. Okay. First of all, I would like to show how to import data into CoolMap. So just click File, Import Data. I'll just import the format as numeric TXT. I actually got some random data here. It's just for the demo. It's just means nothing, but yeah, it's just random data. So as you can see, now the first time you import a data matrix into CoolMap, it's just a heat map. So with the column labels and row labels, but for now the column ontology and the row ontology is just empty. It's just the leaf nodes. Okay, from here I would like to show some basic operations on CoolMap. Okay, first, I would like to show some manipulation operations, and like sorting. Let's say sometimes user want to sort all the rows according to based on the select columns. Here we got a lot of genes in, on the column side. I'll just random choose 3, and by right-clicking on this panel, select sort rows based on select columns. So this is basic for sorting rows, but you have to specify which column you based on. Now, because we have selected 3, we can add another 2 levels. So for the first level, as you can see here, it is actually exactly the same with you sorting a table in Excel. So it's just the first column, the first level is based on column CSPG4, and the second one I could choose GPX1 and in a descending order, and the third one is going to be CHPF in descending order. After you specify all the arguments here, you can then perform the sorting. Okay, as you can see here, all the rows have been reordered depending on the orders you specified here. And okay, it's just, yeah. Once if this column, 2 rows has the same value on this column, so they will sort by the second column in a descending order. Yeah. So CoolMap also comes with some undo functions, like let's just undo the sorting. Yeah, so the row nodes go back to the original order. Okay, so this is the so actually this part we call the CoolMap main canvas, where your data will be displayed. Okay, after this, I would like to show creating a new window from the existing view. Like sometimes the researchers might just want to dig into some selected region or interested region of CoolMap of the data you displayed, like this region. I kind of want to create a new view of this. So just go into the right-click pop-up menu and choose using data from the selected region to create a new window, and name it, and then just this will appear under the CoolMap data where this view is. So as you can see, when you click on those views, it actually directs you to the those names. It actually directs you to the actual view of the data. So this is, I mean, especially when you have a lot of data and a lot of windows, and you don't remember the name or something like that, and you want to go into some views, and you can't really just drag from this side. You can just click on this side, and yeah, vice versa. You can also click this side, and it will automatically select the tree node. Okay. Okay, as you can see, CoolMap also comes with a lot of small widgets as show as tabs here, like some kind of searching function that, like, you can type. Let's say I would like to type S2 here. Now you've got all the nodes which match this name. By not right click, but just clicking it, the CoolMap view will automatically select corresponding row nodes, I think. Okay, yeah, because those nodes comes from the row side. Okay, and also sometimes users might want to filter the data they have imported. So let's say now I want to see the values within, maybe 0.1 to 0.5. Okay, I'll add the first field as larger than 0.1, and another field as
0.5, and then just push the refresh. Now all the other like the values within this range will be displayed as the original color, but yeah, others just filled up. Okay, also we have like a radar for the hill view, the overview of the whole data, and yeah, there are just a lot of operations you can do. I don't really have time to go through all of them, like those other options you can do on the pop-up menu here, and also like here. Because I have a lot of other demos in later slides that use this kind of other widgets, so I'll just skip those widgets for now. So okay. Next, I'm gonna talk about some add-on functions of CoolMap. I kind of want to focus on this part is because all the part I just introduced to you guys is developed by Gang, and I just like fixed some bugs in it and some other functions. But for the plugins, all of those plugins here have all been developed by me. So I'm more, I would say I'm more familiar with those plugins, and so I can show you how those plugins works. Okay. So first of all, I've written a RESTful API plugin for CoolMap. For this plugin, it is pretty simple because it is based on Jersey framework. I don't know if you guys know it, but it's just a very simple RESTful service framework for you to use. And I'll give you a very simple example maybe. Like by visiting this URL, when the CoolMap keeps running here, it actually get the current active matrix in CoolMap into a JSON object. Now you can see that it has a row label field and also have value field and the column labels and like the aggregation method. So other applications can access CoolMap, can fetch data, load it in CoolMap, also like do some operations. Because I can't really do operations through a browser because that is post request. So you can, if you are interested in like writing something, work with CoolMap, you can go, when you start CoolMap, you can visit this page, which shows how many APIs we have for now. The thing is, we currently only support like around 10 operations for data fetching operations, but we would definitely add more to support other developers. So next, let's move to another CoolMap plugin. So this is a very common occasion that researchers might want to do some data exchange with other applications because we have a lot of researchers use Cytoscape or IGV, these kind of applications. So you might want to examine the data in CoolMap as well as in Cytoscape. So I wrote this plugin to facilitate this process. This is basically through like mapping the row and column nodes, mapping the name or unique name to Cytoscape or to IGV. For IGV, we even have like a process to control the expanding of the genes to passing the genes to IGV, and you can control the time and this kind of thing. Okay, I'll still walk you through this by a demo. Okay, first I want to show the Cyberscape, how it works with Cyberscape. Okay. So I modified one of the most popular plugin for Cyberscape, which is called Reactome Wi-Fi. They might not use this name anymore, but still starts with Reactome. But I added some functions to it to show for the demo, and this operation actually loads all the pathways on the left side of the panel, and it still like retrieve all the should be FI networks of all the genes, so that after I done this, I would get all the genes into the Cyberscape's network. So it may take a while because it is a lot and it's through network. Now I got all the genes here. I don't really need to layout it because I don't want to see. A lot of time can spend on the layout thing. But from here, let's go back to the original data. Let's say I've chosen like around maybe 30 genes from CoolMap, and under the plugin menu, I would like to map those nodes without the aggregated values. You can still choose with aggregated values into Cyberscape, because Cyberscape has a table for values as well. Now, as you can see from here, those yellow dots are the nodes I passed from Cyberscape, from CoolMap to Cyberscape. Now, because we have already selected all the genes we need from CoolMap in Cyberscape, now we can do a pathway enrichment from the selected nodes. Okay, so after this is done, you can see that under the metabolism pathway, where we have the most hit. Okay, I'll choose this one maybe. Okay. So this one has 6 hits in it, and I also added another function to retrieve the FI network and as well as the diagram of the pathway. Let's run it. Okay, this might be too simple to show the next phase. Okay, I might not use this one. Let's try this one. Yeah, because this diagram is really simple and I can't really choose other genes from it. Okay, now we got this FI network. This is just all the genes has the interactions between genes in this pathway. And under here, I don't know if you guys can see it, but it is on the bottom left. So the purple elements here all have some hit genes. So sometimes you might want to check if the neighborhood elements has the genes from those neighbor nodes has some special values in CoolMap as well. So you might want to map those nodes back to CoolMap. So by selecting all those nodes here, let's try if there are some mapping nodes in CoolMap. I'm not sure about that, but let's just try. Okay, I have to get rid of the current selection. Okay. Okay, this one doesn't really have any. Okay. Let me try another one. I'm not sure yet because I only got like 50 genes in CoolMap site. There might not be any genes can be mapped to back to that CoolMap site. What about this? Oh, it's about the same. Yeah, because this one is only
You can select the purple ones.
purple. Yeah, but there is just like 2 purple ones.
That's
I'm
okay.
not sure if the. Okay, let's just select the purple ones because purple ones comes with more genes. I mean, yeah. Okay, let's do it now to try if the. Yeah, now you can see that those columns will be selected because those nodes are all mapped from Cytoscape side to CoolMap side. By doing this, users can actually, I mean, of course, it's better to have 2 monitors because you work with 2 applications so that you can examine the data side by side, like in CoolMap and in Cytoscape. Because Cytoscape is more often used in like examining networks, but CoolMap is like for matrix and tables and like gene expression data. Okay, so now we have introduced the interaction data exploration in Cytoscape and CoolMap. So the next I'm gonna show you guys is how to examine sequencing data in like IGV as well as using CoolMap on the side. So it is a kind of a common task for researchers to examine the sequencing data on IGV site using the selected genes in CoolMap. So let's try it. Because I don't really have a very high resolution here, so sometimes I can't fit all the windows here. But now I have started IGV. I'll load some tracks into IGV. Okay. I think I got 5 tracks here. Okay, now you see that we have 5 tracks for the genome data. And let's say we want to examine the genes here, the selected region. Okay, by plugging in IGV and set should be map select column nodes to IGV. We actually gives you a dialog to set up how you want to see this process. You can set up the pausing time between two passing. Like I'll just set to maybe 3 seconds. And numbers of genes to send each time. Let's say 5. Okay. Once you start sending genes from IGV side, you can see those 5 genes has already been mapped. And after 3 seconds pausing, it goes to the second 5 genes. Yeah, the thing is, in this kind of situation, the researchers always have a lot of genes, during the analysis. So this is kind of convenient for automate this process, just by setting up. And you can, the thing is, I can't really display to Windows now. You can pause any time you want and even go back to the last set. But yeah, you have to wait for the network because previous and next. Yeah. So this is kind of convenient for researchers to do things in IGV as well. So this is what we have done to help user to do sequencing exploration in IGV. So for now, we only got those two applications that we can work with. But yeah, I mean, there are like other applications we may add to CoolMap in the future. Okay, so let's move to the next plugin. So we call this plugin sample property plugin. So basically, it's just by this plugin we can generate hierarchical concept structures based on the sample's property values. Okay, I'll still show you how it works by a demo. So for sample property data, users do want to, the researchers always want to do some comparisons between the samples by properties. So I got another data set here. This is a real world data. It's just a gene expression data with 8,000 genes. Let's open it now. Okay. So as you can see, we might get, I don't know, maybe 57 samples here. And we do have a sample property table for it. By importing sample property table, you should go to ontology import because we generate ontologies based on the sample property table. So this is the sample description for this data set. Now after we import it, you can see the sample property table is quite simple. It's just for each sample, it comes with two properties. One is tissue and one is genotype. And they have different values for it. So now, actually, we already got the ontologies for the sample property based on the property values because we have 3 distinct values for the tissue and 3 distinct values for the genotype. So we actually ended up having 9 groups, I think. Yeah, 3 multiplied 3. So now we can actually apply this to the column nodes. Okay. Now as you can see, this level of yellow nodes represent the 9 groups. So you can examine those samples side by side because they all grouped by the values. Sometimes the researchers might want to reorder the properties which appears in this tree. Like, let's say users want to group the samples based on genotype first, and then based on tissue. So you can just drag the column of this table to reorder the properties. Now, after you've done this, in the ontology table, you got another ontology called genotype tissue. This is just the order. So after this, you can replace the column nodes you just applied. Okay. Now you can see all the samples have been reordered by the new order you specified. Sometimes this is not enough for the researchers, like just reorder the properties. Sometimes they might want to set the specific values for the property. Like I want to set a group like contains BMI value from like 25 to 30. I don't know. But yeah, you can specify it in sample property table. The thing is we don't really have any numerical values properties here, so we can show that here. But when you click it, you can actually import group settings from OBO file, in which you can write that file yourself, in which you can specify which value belongs to which group. By this, after you import this OBO file, the new group you assigned will be used to generate the ontology. You can still like view the groups and delete columns. Like when you do the viewing groups, it shows actually how you group the current properties. Now, because tissue only has 3 tissue values, distinct values here, so it has 3 groups. But just as I just said, you can regroup it by like assign those 2 together as a group by writing that file. Yeah, this is just the sample property plugin. It actually helps users to quickly. It is just something that helps users can quickly generate sample ontologies or different sample groupings during this kind of, comparative analysis. So actually, we just use the ontology table here. So this is a very important widget because the basic idea of CoolMap, as I introduced at the first, it is heat map plus ontologies. So you can actually access all the nodes in the ontology here, and even after you select it, it shows how this ontology looks like. And you can like replace the column nodes and append it, insert it. Yeah, you can do a lot of things here and like search the nodes you may be interested in. Okay, so we've done with those 2 widgets. Yeah. Okay, I think that's it for sample property plugin. So next we gonna move to what we call R plugin for CoolMap, which we think is maybe the most important one we have developed for now. Okay, so basically our plugin is just when we use CoolMap, we always think it must be very convenient if we can access R, use R on the data imported in CoolMap. So we introduce R into CoolMap by integrating a framework called RServe. Yeah, which you have to. It is just a library. Each time you have to load the library, but you just have to install it for once. So for our plugin, we offers 2 ways of using it. The first one is user can write their own R functions or use the R functions predefined by us. So this is kind of the basic use of the R plugin. We now offers 2 categories for the functions. The first one is analysis function, and the second one is the clustering functions. So I would like to. So the second way is integrated R command line. I will talk about it later. So first, I want to show you how to write a customized R analysis function for sharing. So I can actually open the folder for you guys. So under CoolMap folder, we have this plugin folder, which you can place all the plugins to. And under this, go into our plugin. We have this scripts folder. Under the scripts folder, you see like we have two categories, analysis and classroom. So currently we only have matrix dot. It's just a t-test analysis with, for the matrix. Okay, let's just open it. So every time if you wanted to write a scripts in R which can be used by CoolMap, you can just open the template to see how to write it. But I do want to show you this, how this is written. So basically, for the first part should be some annotations like descriptions and parameters, but those fields are just optional. Yeah, you can just skip those. But if you add this, when we pass the function, we actually pass the functions into a GUI interface where you can set the parameter, the values of the parameters, and also we can show some descriptions and descriptions for the parameters as well. So this is how we define the t-test function. This is just the function name, and this is the function header, and those are parameters. For if you're gonna use matrix in CoolMap, you just left this kind of parameter the name, but not like assign them anything. If you leave a parameter like this, it's just a matrix. We can select it in CoolMap. And for other kind of parameters, you have to specify what kind of values I'm gonna choose from. Or for numerical one, you have to assign a default value for it. So this is just the header, and in the body, in the function body, it's just do things. Do anything you want to do here, and remember to save it into a list by a named list at the end of the function, and so we can access the list you saved here. So now let's try it. So after you've done this, you put this t-test function into this folder, and after you restart CoolMap, go to analysis, choose the function name you just wrote. Let's say t-test. Okay, here we got the t-test GUI interface. So as you can see, we pass the function and pass all the parameters here. Like all the parameters will be listed here. Like for alternative, there were 3 values you specified in the function header. And also in here, this is the place where you can actually import the matrix from CoolMap. So let's say I'm gonna select this region and select the region into it, and okay, so this is just a selected matrix. For t-test, I do need to select like same amount of rows. Not same amount, but exactly the same rows or columns. So I need to do like this. Let's say should be okay. Should be okay. And based on this, so matrix 2 and matrix 3 has exactly the same rows. So we assign those 2 matrix to the parameters x, y, and we do need the result. You have to specify a variable name for the R variable, which you're gonna use to store the analysis result. Let's just call it t-test.result. Okay. Because we're gonna do it by row, we don't have to change the parameter here. So let's say perform analysis. Okay, it's something wrong here. I might not using the same. I don't know. Let's try to select same row. Wait. 4 and 5. Paired. 3. Oh, maybe because it's the paired thing. Okay, yeah, I just accidentally selected that, but for now, you got the results from the analysis. So as you can see here, we got like p-values, right, that researchers might be interested in. The thing is, if you can just examine the values of the p-value on this dialog, only on this dialog, it might be useless because it's just numeric values. But we do offer you an option to highlight those select variable into CoolMap, like because it's by row, so I need to highlight select to row. Okay, now as you can see, the p-values will be mapped to color as well, because people care about the p-value when it's like small, so it's you can just select like set aggregation type, the mean or something like that when it does have some ontology thing here. But the thing is we don't have that so we cannot show that here. Okay. Also, actually, let's say I can do a clustering here. So this is actually another category of the analysis functions you can add to CoolMap. It is clustering functions. So now we have a hierarchical clustering here. Okay, let's do a clustering on the rows. Okay, because it has 8,000 genes, it may take like, yeah, I don't know, maybe 20 seconds. Okay, it's done now. So as you can see now, the rows has been clustered. And because of now, we have a hierarchical structure on the rows. So those values will not be the original, will not be mapped to the original nodes as they were, because now it like under the clustering 7915, there might be a gene has a p-value of 0.63, and it is maybe the max one. And so you can set the aggregation type to minimum. So this value will change to the gene's value, which the gene which has the minimum p-value, so that you can see what's under it. And you might be interested in to expand the node furthermore to see if there is something you want to see. So, okay, because here we got all the ontologies here, so I kind of want to show how we can expand and collapse the nodes, because as I just said, it is 8,000 row genes, which means 8,000 rows here. But as you can see here, after the clustering, it is only like 50 rows here. It is very convenient for you to see in a very high level of the data. Okay. We can also undo this, like it's just selections, but we can, yeah, we can undo the. I don't know why it's not working. I don't know. Let's just move on. So this is just the analysis functions you can add. Are analysis functions you can add to CoolMap, which helps you to do analysis and also do like clustering, this kind of thing. And okay, next I'm gonna show you how to actually access to our terminal. So we do have this. Okay, this is just something like very similar to the R terminal you use every day. So like type anything like assign a value to a variable, yeah, it just appears here, and it comes with a value with 1. And the nice thing is now you can use the command line. You can do the command line thing towards or on the matrix in CoolMap, which is the same as I just said in the previous slide. You can add the select region into CoolMap. Now we got this matrix here, and as you can see in the variables inspector view, all the values we generated in analysis and in the clustering are all here, which you can further use it, like the p-values. Like if you haven't done the highlight thing here, you can now map it to the column or to the row. Okay, the other thing I want to show is like if you do have a bunch of variables here, you can just type in and the name of the variable and it just search the variable list and show the one you might be interested in. Yeah, and you can examine the value as you want. So basically it's kind of similar to the, it's very similar to the R terminal you use, but it does have something different because we can't really manage the output as the way the real R terminal produce. Like if I want to see like some matrix data here, like I got a something like let's say matrix test matrix. It just have like 6 for like it is 16 by 16 matrix. Okay. It's not working.
So you can just use matrix 6.
Oh, okay. Yeah, the thing is I think it's just smaller to show. I mean, yeah. Yeah, the thing is now you can see we cannot really format the output for now because we just output all the values in a single line. But in the standard R terminal, you can have a very nice view of the output. But yeah, we'll keep working on it. Yeah, I don't know if it's gonna be true, but yeah, it's just here. Okay, the other thing we can do here is like we just have a matrix here called matrix test, and anytime you wanted to examine this matrix in CoolMap, just select here and send the selected to CoolMap. As you can see here, now the matrix is in CoolMap because we all have same value here, so it's just the same color. Yeah, but it's kind of convenient if you produce and generated a lot of matrices here, and you can just map it here. Okay, so it's what I got to say for the R plugin.
We are over the hour, so.
Oh, I'm sorry about
wrap
that.
up
I didn't.
soon.
Yeah. Okay. Yeah. Actually, it's already done. Yeah. Okay. So, any questions? You can just visit the website and download it and try it. And yeah, have any questions? You can just ask me or yeah. Okay. Thanks. I'm sorry about the time. Yeah, I didn't really pay attention to that. Sorry about that. Or yeah, I don't know if anybody have questions, but it's already over time.
In terms of your ontology part, so for example, if you want to show a part of, say, an ontology, a gene ontology, for example,
Yeah.
can you
Load it. Yeah. The thing now, we only support OBO file and 2-column format. So let's say you can import ontology. By simple 2-column, it's just a parent-child relationship age row, and then you can put it into a file and load it to the format, which will automatically generate like for gene ontologies.
okay? So the 2-column is
Yeah, parent-child, yeah, relationship. Yeah. Yeah.
So what about if one parent has multiple child? So you have to.
Yeah, that's fine because multiple child. The thing is, we try to.
Yeah. So
Right.
yeah. So I think so actually it supports like 3 formats. So
3?
yeah. So
Okay. I thought it's just 2.
so GMT is kind of like simple ontology produced by the bloating. So it's basically a categorical name and a list of in the same line. So
paper figure to
So three columns. One is the label.
yeah, one is like the category name. The other is so each line start with like the category or ontology term, and then followed by a list of its members.
Yes.
Oh
Yeah,
yeah.
yeah.
This is
and this
still
one.
like
So for the 2 columns, it's
Maybe
really
more have a
flexible. So
steel.
you like do parent child, you can do multiple